As I was saying in that previous section, epithelial tissue develops from all three primary germ layers. All connective tissue and most muscle tissue derive from mesoderm. Nervous tissue develops from ectoderm. Normally, most cells within a tissue remain anchored to other cells or structures. Only a few cells, such as phagocytes, move freely throughout the body, searching for invaders to destroy. However, many cells migrate extensively during the growth and development process before birth. Cell junctions. The object of this section is to describe the structure and functions of the 5 main types of cell junctions. Most epithelial cells and some muscle and nerve cells are tightly joined into functional units. Cell junctions are connect points between the plasma membrane of tissue cells. Here we consider the 5 most important types of cell junctions: tight junctions, adherens junctions, desmosomes, hemidesmosomes, and gap junctions. Tight junctions. Tight junctions consist of web-like strands of transmembrane proteins that fuse the outer surface of adjacent plasma membranes together to seal off passageways between the adjacent cells. Cells of epithelial tissue that line the stomach, intestines, and urinary bladder have many tight junctions to retard the passage of substances between cells and prevent the contents of these organs from leaking into the blood of surrounding tissue. Adherens junctions. Adherens junctions contain plaque, a dense layer of protein on the inside of the plasma membrane that attaches both to membrane proteins and to microfilaments of the cytoskeleton. Transmembrane glycoproteins called cadherins join the cells. Each cadherin inserts into the plaque from the opposite side of the plasma membrane, partially crosses the intracellular space, the space between the cells, and connects to cadherins of an adjacent cell. In epithelial cells, adherent junctions often form extensive zones called adhesion belts, because these encircle the cell similar to the way a belt encircles your waist. Adherens junctions help epithelial surfaces resist separation during various contractile activities, as when food moves through the intestines. Desmosomes. Like adherens junctions, desmosomes contain plaque and have transmembrane glycoproteins, cadherins, that extend into the intracellular space between adjacent cell membranes and attach cells to one another. However, unlike adherens junctions, the plaque of desmosomes does not attach to microfilaments. Instead, a desmosome plaque attaches to elements of the cytoskeleton known as intermediate filaments and consists of protein keratin. The intermediate filaments extend from desmosomes on one side of the cell across the cytosol to desmosomes on the opposite side of the cell. This structural arrangement contributes to the stability of the cells and tissue. The spot weld-like junction are common among cells that make up the epidermis, the outer layer of the skin, and among cardiac muscle cells in the heart. Desmosomes prevent epidermal cells from separating under tension and cardiac muscle cells from pulling apart during contraction. Hemidesmosomes. Hemi meaning half. Hemidesmosomes resemble desmosomes but do not link adjacent cells. The name arises from the fact that they look like half of a desmosome. However, the transmembrane glycoproteins in hemidesmosomes are integrins rather than cadherins. On the inside of the plasma membrane, integrins attach to intermediate filaments made of the protein keratin. Excuse me, that was a mispronunciation there. On the inside of the plasma membrane, integrins attach to intermediate filaments made of the protein keratin. On the outside of the plasma membrane, the integrins attach to protein laminin, which is present in the basement membrane, which we will discuss shortly. Thus, hemidesmosomes anchor cells not to each other but to the basement membrane. Gap junctions. The gap junctions, membrane proteins called connexins, form tiny fluid tunnels called connexons that connect neighboring cells. The plasma membrane of gap junctions are not fused together as in tight junctions, but are separated by a very narrow intracellular gap, a space. Through the connexons, ions in small molecules can diffuse from the cytosol of one cell to another. The transfer of nutrients and perhaps waste take place through gap junctions in avascular tissue such as the lens and cornea of the eye. Gap junctions allow the cells in a tissue to communicate with one another. In a developing embryo, some of the chemical and electrical signals that regulate growth and cell differentiation travel via gap junctions. Gap junctions also enable nerve or muscular impulses to spread rapidly among cells, a process that is crucial for normal operation of some parts of the nervous system and for the contraction of muscles in the heart, gastrointestinal tract, and the uterus. Epithelial tissue. The objective of this section is to describe the general features of epithelial tissue, list the location, structure, and function of each different type of epithelium. In epithelial tissue, or epithelium, for plural, epithelia, let me repeat that. In epithelial tissue, or epithelium, the plural of which is epithelia, consists of cells arranged in continuous sheets in either single or multiple layers. Because the cells are closely packed and are held tightly together by many cell junctions, there is little intracellular space between adjacent plasma membranes. Epithelial tissue forms covering the lining throughout the body. It is never covered by another tissue, so it always has a free surface. Epithelial tissues have three major functions. Number one, selective barriers that limit or aid the transfer of substances into and out of the body. Secretory surfaces. Number two, secretory surfaces that release products produced by the cell onto their free surface. And three, protective surfaces that resist the abrasive influence of the environment. The various surfaces of epithelial cells often differ in structure and have specialized functions. The apical surface of an epithelial cell faces the body surface, a body cavity, the lumen interior space, or an internal organ, or a tubular duct that receives cell secretions. Apical surfaces may contain cilia or microvilli. The lateral surfaces of an epithelial cell face the adjacent cells on either side. As you just learned and saw, lateral surfaces may contain tight junctions, adherens junctions, desmosomes, and or gap junctions. The basal surface of an epithelial cell is opposite the apical surface, and the basal surfaces of the deep layer of cells adhere to extracellular materials. Hemidesmosomes and the basal surfaces of the deepest layer of epithelial cells anchor the epithelium to the basement extracellular membrane. In discussing epithelia with multiple layers, the term apical layer refers to the most superficial layer of the cell, and the basal layer is the deepest layer of epithelial cell. The basement membrane is a thin extracellular layer that commonly consists of two layers, the basal lamina, that's number one, Lamina. And to repeat, the basement membrane is a thin extracellular layer that commonly consists of two layers, the basal lamina and the reticular lamina. The basal lamina meaning thin layer, is closer to and secreted by the epithelial cells. It contains proteins such as laminin and collagen, which will be described shortly, as well as glycoproteins and proteoglycans, also described shortly. As you have already learned, The laminin molecules in the basal lamina adhere to integrins in the hemidesmosomes and thus attach epithelial cells to the basement membrane. The reticular lamina is closer to the underlying connective tissue and contains proteins such as collagen produced by connective tissue cells called fibroblasts. The basement membrane functions as a point of attachment and supports for the overlying epithelial tissues. The clinical connection. Basement membrane and disease. Under certain conditions, basement membranes become markedly thickened due to increased production of collagen and laminin. In untreated cases of diabetes mellitus, the basement membrane are small blood vessels In untreated cases of diabetes mellitus, the basement membrane of small blood vessels, capillaries, thicken, especially in the eyes and kidneys. Because of this, blood vessels cannot function properly and blindness and kidney failure may result. Epithelial tissues. Epithelial tissue has its own nerve supply, but is avascular. That is, without vascular vessels, without vessels. And they lack their own blood supply. The blood vessels that bring in nutrients and remove wastes are located in the adjacent connective tissue. Exchange of substances between epithelium and connective tissue occurs by diffusion. Because epithelial tissue forms boundaries between the body's organs or between the body and the external environment, it is repeatedly subject to physical stress and injury. A high rate of cell division allows epithelial tissue to constantly renew and repair itself by sloughing off dead or injured cells. Again, a high rate of cell division allows epithelial tissues to constantly renew and repair itself by slogging, slogging, S-L-O-U-G-H-I-N-G, slogging. And I want to repeat that again. A high rate of cell division allows epithelial tissue to constantly renew and repair itself by slogging. A high rate of cell division allows epithelial tissue to constantly renew and repair itself by slogging off dead or injured cells and replacing them with new ones. I hope you don't come across that word again. Epithelial tissue plays many different roles in the body. The most important are protection, filtration, secretion, absorption, and excretion. In addition, epithelial tissue combines with nervous tissue to form special organs for smell, hearing, vision, and touch. Epithelial tissue may be divided into two types. Covering and lining epithelium forms the outer covering of the skin and some internal organs. It also forms the inner lining of blood vessels, ducts, and body cavities in the interior of the respiratory, digestive, urinary, and reproductive systems. Glandular epithelium make up secreting portions of glands such as the thyroid gland, adrenal gland, and sweat glands. Covering and lining epithelium. The types of covering and lining epithelium tissue are classified according to two characteristics: the arrangement of cells into layers and the shapes of cells. Arrangement of cells in layers. The cells are arranged in one or more layers depending upon the function the epithelium performs. Simple epithelium is a single layer of cells that functions in diffusion, osmosis, filtration, secretion, and absorption. Secretion is the production and release of substances such as mucus, sweat, or enzymes. Absorption is the intake of fluids or other substances such as digested foods from the intestinal tract. Pseudostratified epithelium appears to have multiple layers of cells because the cell nuclei lie at different levels and not all cells reach apical surface. Cells that do extend to the apical surface may contain cilia, others Globulate cells secrete mucus. Pseudostratified epithelium is actually a simple epithelium because all its cells rest on the basement membrane. Thus, pseudostratified, pseudo meaning false, false layers. It doesn't have layers at all. They're false layers. They're all rested on the basement membrane. Stratified epithelium, stratum meaning layer. Stratified epithelium consists of two or more layers of cells that protect underlying tissues in locations where there is considerable wear and tear. Cell shapes. The cells vary in shape depending upon their function. Squamous cells are arranged like floor tiles and are thin, which allow for rapid passage of substances. Cuboidal cells Cuboidal cells are as tall as they are wide and are shaped like cubes or hexagons. They may have microvilli at their apical surface and function in either secretion or absorption. Columnar cells are much taller than they are wide, like columns, and protect underlying tissues. Their apical surfaces may have cilia or microvilli, and they often are specialized for secretion and absorption. Transitional cells change shape from flat to cuboidal and back again, as organs such as the urinary bladder stretch, distend, or a large size and then collapse to a smaller size. So once again, transitional cells change shape from flat to cuboidal and back as organs such as the urinary bladder stretch to a larger size and then collapse to a smaller size. Stretch, also known as to distend. Combining the two characteristics, arrangements of layers and cell shapes, the types of covering and lining epithelia are as follows. Simple epithelium. Number 1. A. Simple squamous epithelium. B. Simple cuboidal epithelium. C. Simple columnar epithelium. Non-ciliated and ciliated. Pseudostratified columnar epithelium, non-ciliated and ciliated. Number 2. Stratified epithelium. A. Stratified squamous epithelium, keratinized and non-keratinized. B. Stratified cuboidal epithelium. C. Stratified columnar epithelium. D. Transitional epithelium. Each of these coverings and lining epithelia is described in the following sections and illustrated. The illustration of each type consists of photomicrograph, a corresponding diagram, and an inset that identifies a major location of the tissue in the body. Descriptions, locations, and functions of the tissue accompany each illustration. Simple epithelium. Simple squamous epithelium. The tissue consists of a single layer of flat cells that resemble a tiled floor when viewed from the apical surface. The nucleus of each cell is a flattened oval or sphere and is centrally located. Simple squamous epithelium is present at sites where the processes of filtration, such as blood filtration in the kidneys, or diffusion, such as diffusion of oxygen into blood vessels of the lungs, occurs. It is not found in body areas that are subject to mechanical stress, wear, and tear. The simple squamous epithelium that lines the heart, blood vessels, and lymphatic vessels is known as endothelium. Endo, meaning within, thelium, covering. The type that forms the epithelial layer of serous membranes, such as pericardium, pleura, or peritoneum, is called mesothelium. Middle, meaning meso. Meso, meaning middle. Unlike other epithelial tissues, which arise from embryonic ectoderm or endoderm, endothelium and mesothelium both are derived from embryonic mesoderm. Simple cuboidal epithelium. The cuboidal shape of the cells in this tissue is obvious when the tissue is sectioned and viewed from the side. Cell nuclei are usually round and certainly and centrally located. Once again, cell nuclei are usually round and centrally located. Simple cuboidal epithelium is found in organs such as the thyroid gland and kidneys and performs the functions of secretion and absorption. Simple columnar epithelium, when viewed from the side, the cells of simple columnar epithelium appear like columns with oval nuclei near the base. Simple columnar epithelium exists in two forms: non-ciliated, simple columnar epithelium, and ciliated, simple columnar epithelium. Non-ciliated simple columnar epithelium contains two types of cells: columnar epithelial cells with microvilli at their apical surface and goblet cells. Microvilli are finger-like cytoplasmic projections. Increase the surface area of the plasma membrane, thus increasing the rate of absorption by the cell. Goblet cells are modified columnar epithelial cells that secrete mucus, a slightly sticky fluid at their apical surface. Before it is released, mucus accumulates in the upper portion of the cell, causing it to bulge out and making the whole cell resemble a goblet or wine glass. Secreted mucus serves as a lubricant for the lining of the digestive, respiratory, and reproductive tracts and most of the urinary tract. Mucus also helps prevent destruction of the stomach lining by acidic gastric juices secreted by the stomach. Ciliated simple columnar epithelium contains columnar epithelial cells with cilia at the apical surface. In certain parts of the smaller airways of the respiratory tract, goblet cells are interspersed among ciliated columnar epithelia. Mucus secreted by the goblet cells form a film over the airway surface that traps inhaled foreign particles. The cilia beat in unison, moving the mucus and any foreign particles towards the throat where they can be coughed up and swallowed or spit out. Coughing and sneezing speed up the movement of the cilia and mucus. Cilia also help move oocytes expelled from the ovaries through the uterine fallopian tubes into the uterus. Pseudostratified columnar epithelium. As noted earlier, pseudostratified columnar epithelium appears to have several layers because the nuclei of the cells are at various depths. Even though all cells are attached to the basement membrane in a single layer, some cells do not extend to the apical surface. When viewed from the side, these features give the false impression of multi-layered tissue, thus the name pseudostratified epithelium, pseudo meaning false. In pseudostratified ciliated columnar epithelium, the cells that extend to the surface either secrete mucus, like goblet cells, or bear cilia. The secreted mucus traps foreign particles and the cilia sweeps away mucus for eventual elimination from the body. Pseudostratified non-ciliated columnar epithelium contains cells without cilia and lacks goblet cells and functions in absorption and protection. Stratified epithelium. In contrast to simple epithelium, stratified epithelium has two or more layers of cells. Because of this, it is more durable and can better protect underlying tissue. Some cells of stratified epithelia also produce secretions. The name of the specific kind of stratified epithelium depends on the shape of the cells in the apical layer. Stratified squamous epithelium. Cells in the apical layer of this type of epithelium are flat. Those of the deep layer vary in shape from cuboidal to columnar. The basal, or the deeper cells, continually undergo cell division. As new cells grow, the cells of the basal layer are pushed upward towards the apical layer. As they move farther from the deeper layers and from their blood supply in the underlying connective tissue, they become dehydrated, shrunken, and harder, and then they die. At the apical layer, after the dead cells lose their cell junctions, they are sloughed off, but they are replaced continuously as new cells emerge from the basal layer. Stratified squamous epithelium cells in both keratinized and non-keratinized forms. In keratinized stratified squamous epithelium, the apical layer and several layers deep to it are partially dehydrated and contain a layer of keratin, a tough fibrous protein that helps protect the skin and underlying tissues from heat, microbes, and chemicals. The keratin fills the cells and the organelles die. Keratinized, stratified, squamous epithelium forms the superficial layer of the skin. Non-keratinized, stratified squamous epithelium, which is found, for example, lining the mouth and the esophagus, does not contain keratin. In the apical layer, and several layers deep to it, and remain moist. Let me repeat that. Non-keratinized, stratified squamous epithelium, which is found, for example, lining the mouth and esophagus, does not contain keratin in the apical layer and several layers deep to it and remains moist. The organelle remains in this deep epithelium. Both types of stratified squamous epithelium form the first line of defense against microbes. Clinical connection. Papa-Nicolau. A Papa-Nicolau test, also called a Pap test or Pap smear, involves collection and microscopic examination of epithelial cells that have been scraped off of the apical layer of a tissue. A very common type of Pap smear involves examining the cells from the non-keratinized stratified squamous epithelium of the vagina and cervix, the interior portion of the uterus. The inferior portion of the uterus. This type of Pap test is performed mainly to detect early changes in the cells of the female reproductive system that may indicate a precancerous condition or cancer. In performing a Pap smear, a physician collects cells, which are then smeared on a microscope slide. The slides are then sent to a laboratory for analysis. Pap tests should be started within 3 years of the onset of sexual activity, or age 21, whichever comes first. Annual screening is recommended for females ages 21 to 30 and every 2 to 3 years for females ages 30 or older following 3 consecutive negative Pap tests. Stratified cuboidal epithelium. This is a fairly rare type of epithelium in which cells in the apical layer are cuboidal. Stratified cuboidal epithelium mainly serves a protective function, but it also has a limited role in secretion and absorption. Stratified columnar epithelium, like stratified cuboidal epithelium, stratified columnar epithelium also is uncommon. Usually the basal layer consists of shortened, irregularly shaped cells. Only the apical layer has cells that are columnar in shape. This type of epithelium functions in protection and secretion. Transitional epithelium. Transitional epithelium, a type of stratified epithelium, is present only in the urinary system and has a variable appearance. It is, in its relaxed or unstretched state, transitional epithelium looks like stratified cuboidal epithelium, except that the cell in the apical layer tend to be large and rounded. As the tissue is stretched, the cells become flatter, giving the appearance of stratified squamous epithelium. Because of its elasticity, transitional epithelium is ideal for lining hollow structures that are subject to expansion from within, such as the urinary bladder. It allows the urinary bladder to stretch to hold a variable amount of fluid without rupturing. Glandular epithelium. The function of glandular epithelium is secretion, which is accomplished by glandular cells that often lie in clusters deep to the covering and lining epithelium. A gland may consist of a single cell or a group of cells that secrete substances into the ducts or the tubes, onto a surface or into the blood. All glands of the body are classified as either endocrine or exocrine. The secretion of endocrine glands, endo meaning inside, crine meaning secretion, inside secretion. The secretions of endocrine glands enter the interstitial fluid and then diffuse directly into the bloodstream without flowing through a duct. These secretions, called hormones, regulate many metabolic and physiological activities to maintain homeostasis. The pituitary, thyroid, and adrenal glands are examples of endocrine glands. Endocrine glands will be described in detail in Chapter 18. Exocrine glands, exocrine, E-X-O-C-R-I-N-E, or exocrine glands, exo meaning outside, crine meaning secretion once again. Exocrine glands secrete their products into ducts that empty onto the surface of a covering and lining epithelium, such as the skin surface of the lumen of a hollow organ. The secretions are exocrine glands. The secretions of exocrine glands include mucus, sweat, oil, earwax, saliva, and digestive enzymes. Examples of exocrine glands include sudoriferous glands, which produce sweat to help lower body temperature, and salivary glands, which secrete saliva. Saliva contains mucus and digestive enzymes, among other substances. As you will learn later in the text, some glands of the body, such as the pancreas, ovaries, and testes, are mixed glands that contain both endocrine and exocrine tissue. Structural classification of exocrine glands. Exocrine glands are classified as unicellular or multicellular. Exocrine glands are classified as unicellular or multicellular. As the name implies, unicellular glands are single-celled. Goblet cells are important unicellular exocrine glands that secrete mucus directly onto the apical surface of a lining epithelium. Most glands are multicellular glands, composed of many cells that form a distinctive microscopic structure or macroscopic organ. Examples include sudoriferous, sebaceous, and salivary glands. Multicellular glands are categorized according to two criteria. 1. Whether their ducts are branched or unbranched, and 2. The shape of the secretory portions of the gland. If the duct of the gland does not branch, it is a simple gland. If the duct branches, it is a compound gland. Glands with tubular secretory parts are tubular glands. Those with rounded secretory portions are acinar glands, or acinar. Acin equals berry. And these glands are also called alveolar glands. Tubuloacinar glands have both tubular and rounded secretory parts. Combinations of these features are the criteria for the following structural classification scheme for multicellular exocrine glands. Simple glands. Simple tubular. Tubular secretory part is straight and attaches to the single unbranched duct. Example, glands in the large intestines. B. Simple branched tubular. Tubular secretory part is branched and attaches to a single unbranched duct. Example, gastric glands. C. Simple coiled tubular. Tubular secretory part is coiled and attaches to a single unbranched duct. Example, the sweat gland. Simple acinar. Secretory portion is rounded and attaches to a single unbranched duct. Example, glands of the penile urethra. Simple branched acinar. Rounded secretory part is branched and attaches to a single unbranched duct. An example would be the sebaceous gland. 2. Compound glands. Compound tubular. Secretory portion is tubular and attaches to a branched duct. An example would be bulbourethral or Cowper's gland. B. Compound acinar. secretory portion is rounded and attaches to a branched duct. Example, mammary glands. Compound tubuloacinar. Secretory portion is both tubular and rounded and attaches to a branched duct. Example, acinar glands of the pancreas. Functional classification of exocrine glands. The functional classification of exocrine glands is based on how their secretions are released. Secretions of merocrine glands, mero meaning a part, are synthesized on ribosomes attached to either endoplasmic reticulum, processed, sorted, and packaged by the Golgi complex, and released from the cell in secretory vesicles via exocytosis. Let's read that again. Functional classification of exocrine glands. The functional classification of exocrine glands is based on how their secretions are released. Secretions of merocrine glands are synthesized on ribosomes attached to rough endoplasmic reticulum, processed, sorted, and packaged by the Golgi complex, and then released from the cell in secretory vesicles via exocytosis. Most exocrine glands of the body are merocrine glands. Examples include the salivary glands and the pancreas. Apocrine glands accumulate their secretory product at the apical surface of the secreting cell. Then, that portion of the cell pinches off from the rest of the cell to release the secretions. The remaining part of the cell repairs itself and repeats the process. Electron microscopy has recently confirmed that this is the mechanism of secretion in the mammary gland. Recent evidence reveals that the sweat gland on the skin, named apocrine sweat glands, after this mode of secretion actually undergo merocrine secretion. The cells of holocrine glands accumulate a secretory product in their systole. As the secretory cell matures, it ruptures and becomes the secretory product, a secretory product. Then sloughed off, the sloughed off cell is replaced by a new cell. One example of a holocrine gland is a sebaceous gland of the skin.